So the last thing I'm going to talk about is some visualizations. So with the new Nephele 2.0, we tried to make visualizations that would help our users be able to do analysis and edit them themselves rather than for them to be somewhat static. So we added these interactive heat maps. So the heat map is made by a program called Morpheus. It's actually also a web app that you can use by the Broad Institute. And on our website in the tutorials, we have some information about how you can do some common tasks for our specific output, right, for the taxonomic output of Nephele. But we also have a link to the documentation from the Broad's website, the Morpheus documentation. And if you're at all interested in using these heat maps, which you should be, I think they're really cool. They allow you to do a lot of stuff. You should really go and read. You can really, through the tutorials and the documentation on the Morpheus website, because it's pretty powerful in a lot of the things that you can do. You can you can change not just how it looks visually, but you can do some statistical analysis with some hierarchical clustering. I think under this nearest neighbors, it tells you how to do some marker selection, so you can look for differentially abundant taxa between groups and run some, do some chi-squared tests and other kinds of statistics as well. And you can do it all in the, in the web page, the standalone HTML file itself. The other thing we added were these graphs that were made with the Plotly JavaScript library. So on our website, we have some extremely short videos that show how you can do very simple edits to the graph itself. But for bigger edits, this is a busy slide, but I just wanted to show what was possible. If you click on this export data to Plotly, that will open up the data set in the Plotly Chart Studio. And you can do lots of different kinds of edits. In this case, if you wanted to change some colors, you can also choose what you'd like to plot. Like we usually plot in our standard plots the first and second principal components, but you could potentially plot any of the other ones as well. If you wanted to, we by default color by the treatment group and our output, but if you wanted to color by a different column in your mapping file, you could, I think this transform section has an option called split, and so you can, what they would consider split on a different column. So here I've chosen to split on a description column instead, and you can color differently. And if you want to choose the specific colors that you want, I think under style, that helps you style the plot a little bit. If you go to traces, you can choose either one of the palettes that they have, or you can choose the exact color you want individually. And they also have lots of different plot types. You don't just have to do this scatter plot. I mean, once you export the data to the grid, you can do anything with it. You don't have to stick to that. And here's like a 3D scatter plot that I made just using this free version of Plotly. And they have a lot of tutorials that show how to do all of these plots on their website as well. And so hopefully this allows you to make figures for publication a little bit more easily than you could before. So if you want to try it out yourself, we have some example output files on our tutorials page. If you go to the tutorials page and you scroll all the way down to the bottom, you can download some example graphs and try out some of the tutorials from the Plotly and Morpheus sites as well. So that's all we were going to talk about in this webinar. I think almost all of the things that I was just going to say one thing, which is I think almost all of the things that we talked about today are actually in different pages on our website, like our FAQ page and on the tutorials and on the individual pipeline pages. So I encourage you to go and read those as well.